We do have 2 more minutes, yes. We're on the new universal Harvard time. We used to wait until 7 minutes after anything.
Yes.
But now we have to start right on time. So
We've been like that for a long time because there's somebody coming in after you, and you got to get out.
That's right. We have a 15-minute window to clear this.
That's what we have. Yeah.
So my introduction will be brief. I don't think I'll steal much time from you. And people, I guess at 1 o'clock it'd be good if, I don't know, you've already moved on to Q&A or something like that.
Yeah, okay.
So,
Yeah, this normally takes me about 45-ish minutes,
that's probably
50, depending on.
And Sage gave you, okay, you've got all.
Laser pointer, water, backup laser pointer.
Okay, I think I'm going to get started hewing to the new universal FAS schedule. I'm going to yes. Use the podium, please, Mike. All right. So I'm going to start on time, uncharacteristically on time, but we're all trying to get used to this now. So welcome to the first CBS seminar of the new academic year. I'm very happy to introduce Kate Wassum to you. Kate got an early start doing research. She was an undergrad at UNC, and she found something that she loved, and she really has stuck with it, I think, throughout her career. As an undergrad, she worked on the effects of dopamine in nucleus accumbens and how addictive substances like cannabinoids affect it. And after finishing her undergraduate degree, she made a fateful decision to go to UCLA for her PhD. And she's never left. She got her PhD there, she did a brief postdoc there, and now she's a tenured faculty member there. And her work has been on a variety of aspects of mechanisms underlying how animals learn when they're seeking reward. And when computational modeler says reward, we write R, or at any given time there's an R of t or something like that. Of course, for an animal, it's much more complicated, and her work has been to try to figure out anatomically what's going on, neurochemically what's going on, the mechanistic underpinnings of it. And in particular, I think that today she will be addressing the fact that we have to, as animals, remember past rewards and recall them, retrieve those memories when we need to. All of that, of course, at the basic mechanistic level is poorly understood. And so I'm looking forward to hearing what Kate has figured out so far.
Thank you so much for that very nice introduction. All right, yeah, so I'm going to talk to you today about how the cortex, the orbital frontal cortex in particular, and the amygdala interact to inform decision making. And when we're making a decision, essentially what we do is we get into the time machine that is in our brains and we use it to take ourselves into the future. And that allows us to evaluate the potential outcomes and consequences of the different options that we have available to us. And that time machine is programmed by our memories. What's happened in the past informs what we think is going to happen in the future. Thus, I've been particularly interested in understanding memories, and especially of rewarding events, how those memories are formed and stored in the brain, how they get retrieved under the right circumstances to influence what we think is going to happen in the future so that we can make smart decisions. And I'm interested in this from a very basic science point of view, but also because we know that decision making can be disrupted in a variety of different mental illnesses. One that we're kind of keen on is addiction, wherein we know that there can be deficits in how memories of rewarding events are formed and stored in the brain. There can be deficits in how this information is retrieved. Maybe it's not retrieved under the right circumstances and things like this. And of course, there can be biases away from decision-making strategies in favor of more automatic behavioral control strategies. And so we've been working, and all the data I'm going to talk to you, most of the data I'm going to talk to you about today are from sort of the basic science level of trying to figure out what circuits in the brain support the formation of reward memories. And I'm going to tell you about two types of memories. The first that I'll focus on is the value of rewarding events, and then later on we'll get into how rewarding events are linked up with stimuli in the environment that predict them. So for the first half of the talk, I'm going to focus on something that's exemplified, something we all do on a daily basis. It's lunchtime, you're hungry, you've got to get something to eat. And one thing your brain might do, as I just said, is sort of think about all of the different options that you have available. And in order to select between these options, you'll have to consider their value. And not sort of their static or absolute value, but their value to you in your current state of hunger. So, for example, coffee may be highly valuable when it's 7 in the morning and you didn't get much sleep last night and you've got a grant to bang out. But it's going to be less valuable when it's lunchtime and you're hungry. In that case, items that have calories and nutrients are going to be more valuable to you. And these values are something that we know that we learn. So if, for example, this fellow has previously gone to In-N-Out, a very popular hamburger chain in Southern California, this would be an excellent opportunity for him to learn the high value of hamburgers in the hungry state, such that when he's hungry again in the future, he could retrieve that information and may be more likely to select a hamburger over other food items for which he's not had that experience, or other items that he knows are just not quite as valuable to his current state of hunger. And we've been sort of talking about this as reward value encoding versus reward value retrieval and implementation for decision making. Now, to model this in the laboratory, we simplify things way down, and we look at the decision to engage in a lever press. So we take rats and we train them to press on a lever to receive a sugar reward. And they're trained only deprived of food for a few hours. And in this state, the sugar it's okay. It's got a fairly low value, and it supports a fairly low rate of lever pressing. We can then take the animal and make him very hungry, but we'll find that he won't actually change his rate of responding on the lever. So the 100% baseline here is how rapidly the animals were responding for the sugar when they were sated. But we've made the animal hungry, and he actually doesn't shift his response vigor away from that baseline. But if we allow the animal to experience the sugar hungry, this will be what we call a value encoding or an incentive learning opportunity, a chance for him to learn how valuable sugar is when he's hungry, so that if we bring him back in the future, he can now retrieve this memory, and he will appropriately escalate his reward-seeking actions for that particular reward. And we'll find things like how palatable he found the sugar the last time he experienced it. As best we can sort of measure it by looking at the licking responses, will positively correlate with how vigorously he'll pursue the reward. So that's the basic task. Animals are trained sated to lever press for sugar. They're then allowed to experience the sugar hungry for the first time. This is done totally non-contingently to avoid any reinforcement-related confounds or any caching of value to the action itself. So the animal's just experiencing the sugar and learning of its high value. Controls just experience the sugar in the very familiar sated state. Then all the rats are made hungry, and they're allowed to press on the lever in a brief extinction test, and this we refer to as the retrieval phase of the experiment. So Melissa Malvallas is a really talented now project scientist in my lab who's been working on these projects, and she and I were curious what circuits in the brain might be important for supporting the encoding and retrieval of reward value. And we honed in on the basolateral nucleus of the amygdala as perhaps being a key locus for at least maybe encoding these reward values. And as a first pass to test this hypothesis, she took glutamate biosensors that make fairly close to real-time measurements of glutamate release in awake behaving animals, and she recorded glutamate release in the basolateral amygdala during the value encoding phase of this experiment. And here what you'll see is two examples. So this rat here is going to experience the sugar hungry for the very first time, so encode something new about its value. And this is our control rat here. He's just experiencing the sugar in a very familiar sated state. You'll see the glutamate response go down below. And there you can see they just poked their nose into the port. And in this guy, he's tasting the sugar, he's learning of its new high value, and that's associated with this surge in glutamate release in the basolateral amygdala. Whereas this rat, he's also experiencing the sugar, but we don't see that same increase in glutamate release in the basolateral amygdala. You can see it again when I show you the data collapsed across trials and across animals. We get this nice transient elevation in glutamate release in the basolateral amygdala when animals are learning something new about the value of a sugar reward, but not otherwise when they're just experiencing that reward and not learning anything new. I find it kind of interesting that early on in this 30 trial session, when you would expect learning to be maximal, is when this signal is also maximal, and then it declines over trials on the session. So that suggested to us that glutamatergic activity in the BLA is tracking the encoding of reward value. We did some pharmacology and also found that it was, in fact, also necessary for encoding reward value, and in particular at the NMDA receptor. So the BLA glutamatergic activity there seems to be tracking the encoding of reward value, and that suggests that there would be an excitatory input signal to the BLA that would be supporting it in this function of encoding value. And for this, we turn to the orbital frontal cortex. We know that it sends a dense glutamatergic projection into the BLA, and it has itself been implicated in value on occasion. Melissa did some tracing work, and she found that both the lateral and medial, and the medial is also a bit more anterior, subdivisions of the orbital frontal cortex send dense excitatory projections into the BLA, and because we wanted to maximize our chances of getting a positive result, we independently targeted both of these projections. Of course, to do this, we're using optogenetics and chemogenetics, which we validated now a few different times in the lab, in this case using slice physiology, and in this case in vivo using glutamate biosensors. In both experiments, we co-expressed channel rhodopsin and the inhibitory dread receptor HM4DI in the orbital frontal cortex, and we wait 8 weeks for that to traffic to the terminals, wherein we find that we can optically evoke glutamate release that we can then prevent ourselves from evoking by applying CNO either to the bath or locally infusing it. All the manipulations I'm going to talk to you about today are terminal manipulations. So armed with these tools, Melissa went on to ask, do you need projections from the orbital frontal cortex to the basolateral amygdala to encode a positive shift in a reward's value? To do this, after training rats sated to lever press for sugar, they all get to experience the sugar hungry, and in some of them we were going to inactivate lateral OFC to BLA projections. And during that test, we saw no effect online. It didn't make them check any differently for the reward. It didn't make them find the sugar to be any more or less palatable. But when we tested them drug-free the next day, we found evidence that they had not actually encoded the upshift in the reward's value. So whereas controls, after experiencing the sugar hungry, will appropriately escalate their pursuit of that sugar, pressing on the seeking lever more, if we had inactivated lateral OFC terminals in the BLA during the value encoding opportunity, they were unable to do this. We saw no effect when we inactivated medial OFC to BLA projections, suggesting to us that lateral but not medial OFC to BLA projections are necessary for encoding reward values. And we suspected that the activity in projections from the lateral OFC to the BLA may be in part sort of conveying a value signal into the basolateral amygdala. And to test that idea, we turned to optogenetics and we decided, okay, we'll stimulate these projections from the lateral OFC to the BLA, and we glutamate conditions in which there would be no such signal. So animals, after being trained to sugar in the familiar low-value sated state again, except now every time they taste the sugar, we're stimulating lateral OFC terminals in the basolateral amygdala. And again, that didn't do anything online. Didn't make them go back and check for the reward anymore. It didn't make the reward any more palatable. But when we tested them the next day, in this case, no stimulation is happening. They're just allowed to press on the lever. We saw this really robust increase in their pursuit of this sugar reward, as if they found the sugar to be more valuable than it really was, than the controls would say it was, and they were pursuing it with greater vigor. And to convince ourselves of that, we did a variety of controls. In this case, what we did was an identical stimulation pattern. So they're in the same place. The stimulation is paired with a food item, but it's just a different food item than they were going to be working for the next day. It was actually a food pellet. And under these circumstances, we didn't see that same elevation in reward-seeking activity, which suggests it's not as if we had made the context more excitatory or something like this, but rather these data indicate to us that the stimulation had instantiated value to this specific sugar reward that the animals were now working to pursue. And we found it quite interesting that the magnitude of this increase completely mimicked the effect of an authentic incentive learning opportunity that would be brought about by actually experiencing the sugar hungry. So these data suggested to us that projections from the lateral OFC to the basolateral amygdala are both necessary and sufficient for encoding the value of a rewarding event. But we suspected that the BLA might be doing a little bit more than just encoding information about rewards. Certainly the basolateral amygdala has a long history of being thought of as an emotional learning center in the brain. That's been really well borne out in terms of fear conditioning and for aversive events, but also for rewarding events. But we suspected that the BLA might not just be doing things about learning about rewards or aversive events, but that it might also be important for retrieving this information. And if that's true, then it's also critical for implementing decisions, or at least contributing to the decision-making process. And to test this, we went on in our subjects that we had the biosensors implanted in, and we made glutamate measurements during the value retrieval phase of the test. And so you'll see that in this rat here. So this guy here, he's hungry at test, and he's previously experienced the sugar hungry, so he can retrieve that information to guide his decisions about pressing on the lever. And there you can see this transient elevation in glutamate release just prior to the rat engaging in a bout of reward-seeking activity, not really prior to each individual action, but prior to these bouts of reward-seeking. And there's another one just there prior to another bout of reward-seeking activity. Now this guy here is also hungry, and he's also going to be pressing the lever, but we haven't let him taste the sugar hungry before, so he doesn't actually know how good it is while he's hungry. And you can see here, he's just about going, he's going to press on the lever, and we see no such fluctuations in glutamate release in the basolateral amygdala occurring then. These data look really nice when we collapse in glutamate release immediately prior to bouts of reward-seeking activity, which is interestingly the time at which you would expect the animal would be contemplating the consequences of his actions and their value and these sorts of things, right before he's engaging in a bout of reward-seeking. And this only occurs in animals who have learned and thus can be retrieving the value of the reward they're working for at the moment. And we can look at this in a variety of different ways showed here, but the point is that the magnitude of this signal predicts on a trial-by-trial basis how vigorously the animal will pursue the reward, how long he will stay on the lever, how rapidly he will press on the lever, and these sorts of things. So these data suggest to us that glutamatergic activity in the basolateral amygdala may be tracking not only the encoding, but also the retrieval of reward value information. Also, if you inactivate glutamate receptors in the BLA, you can prevent this kind of value-guided reward-seeking activity. So then, after having found this sort of information and knowing that the lateral orbital frontal cortex was important for encoding reward value, we thought, okay, it's probably also that these projections are active again for the retrieval of this information. And that beautiful hypothesis was debunked when we did the experiment where we inactivated now lateral OFC terminals in the BLA, not during the value encoding phase, but rather during the retrieval phase of the test. We saw no effect of inactivating lateral OFC to BLA projections. But where we did find an effect was when we inactivated now projections from the medial OFC to the BLA. If we allow animals to experience the sugar hungry, encode its value, and then ask them to implement that information for their lever pressing activity, under these conditions, if we inactivate medial OFC to BLA projections, we strongly attenuate their behavior. This suggested to us that activity in medial OFC to BLA projections might be necessary for retrieving reward value. And if that's really true, if we were to inactivate these projections under conditions in which they're otherwise engaged in lever pressing but not having to retrieve reward value from memory, then we should see no effect. We tested that in two ways. The first way we did this here is simply to make them hungry and let them lever press, but to omit the value encoding opportunity. So their lever pressing here can't be driven by any retrieval of a reward value information because we never let them learn it. And under these circumstances, we see no effect of inactivating medial OFC to BLA projections. We went on in these subjects and we let them actually experience the sugar, learn about its value in the hungry state, but we didn't require that they retrieve that information from their memory because we actually made the reward present at test. So they were able to, after pressing, collect the reward, hold its value in memory, working memory, long enough to then engage in another bout of reward seeking. And under these circumstances, if we inactivate medial OFC to BLA projections, we see no effect. And that allowed us to hone in on the idea that activity in the medial OFC and its projections to the BLA is important for retrieving reward value information for use in making adaptive reward pursuit decisions. To test that idea a little bit further, Melissa Malvayas teamed up with Michael Murphy in the lab to ask whether if they could stimulate these projections, could they help the animal to retrieve a reward's value under circumstances in which it might be normally difficult for them to do so. To do that, they had to design a behavior which we've now called sort of a sub-threshold incentive learning or sub-threshold value encoding procedure. Essentially what they did was, after training animals sated to lever press for sugar, and I've not yet told you that sated is actually deprived of food for 4 hours prior to the test, they allowed the animals to experience the sugar following 8 hours of food deprivation. So a little bit more hungry, kind of a moderate hungry state. And then they tested the animals in that same 8-hour food deprived state the next day, and in controls, they don't actually change their responding that much from the baseline level. So we think perhaps it's difficult for them to disambiguate the value of the sugar when they're a little bit hungry versus a little bit less hungry. It's maybe a little less clear for them. But if during this test we stimulated medial OFC terminals in the BLA, they were able to appropriately upshift their behavior. And this entirely depended on them having previously received this learning opportunity, this sort of sub-threshold learning opportunity. If we omitted that, the stimulation did nothing. It also did nothing if the value of the sugar was well learned and low, or if it was maximally learned as a result of experiencing the sugar in a very hungry state. So these data suggested to us that whereas projections from the lateral OFC to the BLA are important, necessary, and sufficient for encoding information about a reward's value, projections from the medial OFC are necessary and sufficient for retrieving that information under the right circumstances, implementing that for use in making decisions. And so after having sort of found these results, we started to become curious whether this architecture, medial OFC for retrieval, lateral for encoding, or lateral for learning, medial for memory, one of my colleagues sort of came up with that, whether this would apply to other forms of reward memory. So if we go back to our lunchtime example, oftentimes what happens, you might be sitting in your office, there's no food cues around, you're kind of thinking about what options might exist in your environment, like I described. But another common thing that happens to me quite frequently, is you're in your office, or maybe you're in the lab, and your lab mate comes in with this pizza box, the remnants of the pizza that they had for lunch. And that can act as a stimulus, makes you think about pizza, how good it might be right now, and that might bias your choice of lunchtime options to going to get some pizza or something like this for lunch. And this relies on what we would call a stimulus outcome or a stimulus reward memory. In the past, you've gone to the pizza place, you've seen the pizza logo, the pizza box, you've had some pizza, you've linked these two up. It's a stimulus that's associated with a specific reward. And now when you see this pizza box in the future, it can make you think about pizza and influence your decision making in that way. So we're curious whether the same architecture, lateral for encoding, medial OFC projections to the BLA for retrieval, might also be important for encoding these stimulus reward memories. Now, to test this in the lab, we use a couple of different behavioral paradigms. The first one is Pavlovian to instrumental transfer, the specific variety of that task, wherein rats get two different cues, a white noise and a tone, and they're paired with two different rewards, so like white noise pellets, tone, sugar solution. And after that Pavlovian conditioning, they then get instrumental conditioning, where there's two levers, and pressing on one of them gets them pellets, and the other one will get them sugar solution. And then we put the animal in a choice for the first time at test, where both levers are available and they can press on them at will, and most of the rats like pellets and sugar fairly similarly, so they'll press on them fairly equally. But then periodically through the test, we'll play the cues, and we'll find that when, for example, we play the sugar cue, the animal will increase his responding, and he'll do so selectively on the lever that he previously had associated with sugar. And when we play the pellet cue, you'll get the opposite effect. His lever pressing will increase, and it will do so selectively on the action that had been previously associated with pellets. A pattern of behavior like this tells us a lot of different things. It tells us that the animal has learned and can retrieve the stimulus-outcome relationships, that he has learned and can retrieve the action-outcome relationships, and that he's able to use those memories of the rewarding events to appropriately guide his decision-making. As a secondary task, we use a devaluation procedure. Often this will be in the same animal, so they're trained identically, 2 cues, 2 rewards, 2 levers, 2 rewards. And then we do a devaluation procedure where the animal is allowed to eat as much as he wants of one of the food pellets, one of the outcomes. In this case, he's consuming to satiety the food pellets, rendering them temporarily devalued. And the test here is quite simple. He's just in the box, and there's a food port sort of available, and we're monitoring how much he's going into that food port. And when we play the cue of the valued item, in this case the sugar, the animal will sort of think about the sugar, and he'll go to the food port. If it's got a good value at the moment, he'll go to the food port quite a bit. But when we play the cue of the devalued outcome, the animal will withhold going to the food port quite as much. And a pattern of behavior like this tells us first that the animal knows the unique stimulus-outcome relationships, and secondarily that he knows the value of each outcome at the moment. So we pair these two tests up together in order to assess how stimuli in the environment, by way of their relationship with rewarding events, can influence decision-making and adaptive behavior. So Ash Morse, when she was in the lab, asked, do you need the basolateral amygdala to encode these kind of stimulus outcome memories? And she asked that by inhibiting the BLA during Pavlovian conditioning, and we did this optogenetically so that we could time that inhibition specifically during the cue when the reward was being experienced. So the cue's on for 2 minutes, during which the outcome is delivered on average every 30 seconds, and every time the outcome was delivered and the animal went to consume it, she inactivated the BLA. And it turns out that you don't need the BLA to form a simple Pavlovian conditional response. This is their food port approach responding. They'll go to the food port during the cue before the reward comes. Actually, even very well over training, they look no different than controls. But when she used these two tests to probe the content of the learning, what did they actually learn during the Pavlovian conditioning? Whereas the controls really nicely learned the unique stimulus-reward relationships, if she had inactivated the BLA, she prevented them from forming those associative memories. And so now at test, there's actually no inhibition happening. We're just sort of assaying what they had learned during the training, and the controls, as you can see here, are appropriately increasing their responding during the cue on the action that earns the same thing as the cue, telling us that they know the cue reward relationships, but those animals who got BLA inactivation during Pavlovian conditioning are not capable of doing that. And when we look at the devaluation test, we get a similar pattern of results, whereas controls are appropriately able to adjust their conditional food port approach responding based on the value of the reward that's being predicted. If we had inactivated the BLA during conditioning, they can't really do that. And that tells us that the basolateral amygdala is necessary for encoding relationships between stimuli in the environment and the unique rewarding events with which they are associated. And based on our findings that the lateral OFC to the BLA was important for encoding reward value, we suspected, well, maybe you need those projections also to form these stimulus outcome memories. And so Ash did the experiment again, except this time she put the ARCH-T virus in the OFC with her fiber over the BLA. And she got a very similar pattern of results. They're still able to form a basic Pavlovian conditional response, but when we challenge that learning, when we evaluate the content of that learning, she found evidence both in the Pavlovian instrumental transfer task as well as in the devaluation task that they had not actually formed the unique stimulus-reward relationships. So these data tell us that projections from the lateral OFC to the BLA are important for encoding stimulus outcome memories. Now the BLA, we think, is not just important for encoding this information, it's also important for retrieving this. And we think this based on our evidence that if we make recordings of glutamate release in the BLA during these Pavlovian instrumental transfer tasks, we will again see surges in glutamate release that occur prior to bouts of reward-seeking activity, and they are again selective. In this case, when the cue comes on, and the animal goes to press on the lever that earns the same thing as the cue predicts, as if the cue is making him think, oh, pellets might be more available right now, they're good, I'm going to go press for them. Those bouts of reward seeking are preceded by glutamate release events in the BLA. But when the cue comes on and the animal goes to press on the other lever for whatever reason, we don't see those same release events, and again you'll see that the magnitude of this will predict the vigor of reward seeking. And if you inactivate the basolateral amygdala, you will prevent their ability to use cue reward memories to influence their decision making. So this sort of led us into the idea that, well, perhaps the medial orbital frontal cortex may be again facilitating the BLA's function in retrieving reward memories, this time stimulus outcome memories. Nina Lichtenberg has been heading up these projects and a few of the next ones that I will tell you. And here what she did was chemogenetically inactivate medial OFC to BLA projections, this time during the sort of the retrieval phase of the experiment, during the test. So they learned everything up manipulation free, and then she inactivated or not medial OFC terminals in the BLA prior to the Pavlovian to instrumental transfer or devaluation tests. And what she found here is that whereas controls are really well capable of using the cue reward memories to guide their choice behavior, if she inactivated medial OFC terminals in the BLA, they're not really capable of doing that, nor are they capable of using the current value of the predicted reward to guide their behavior. And this pattern of results suggested to us that they're really just not able to retrieve the appropriate reward memory given the stimulus that they're being presented with. So here we've again sort of working on that same architecture suggested that projections from the lateral OFC to the BLA are important for encoding stimulus reward memories, whereas those from the medial OFC to the BLA are important for retrieving these memories. And putting these two things together, first data set and the second data set I was talking to you about, we've kind of getting to the idea that projections from the lateral OFC to the BLA may be important, perhaps more broadly, in encoding memories of rewarding events. Memories that we think in part may be stored in the BLA for reasons that I'll get to. Whereas the medial OFC to the BLA projections, these might be important, their activity might be important for retrieving the right reward memory given one's circumstances. And those circumstances could be defined internally based on your hunger state, and knowing the value of an anticipated reward based on your hunger state. But it also may be defined by stimuli in the environment telling you what rewards are more or less likely to be available. So these projections may, we think, be sending information about the current state to the BLA to retrieve the right memories for your exact circumstances. Now, of course, the next question is, where is this information going after the basolateral amygdala? And probably the answer is to a lot of places, but in part we think it might be going right back to the orbital frontal cortices. And we think that because we know that the BLA projects, in this case I'm showing you to the lateral orbital frontal cortex, doing some tracing. Nina found that BLA sends a really nice projection into the sort of lateral ventrolateral orbital frontal cortex, and those projections are themselves activated by stimuli in the environment that predict rewarding events, in this case doing it kind of crudely by looking at an immediate early gene marker. Let's see. Oh, there we go. And sort of to test the idea that these projections might be important for retrieving stimulus outcome memories, what Nina did was to use a chemogenetic approach to inactivate now BLA projections to the lateral orbital frontal cortex, so sort of back projections. And what she found was that when she did that inactivation, either prior to a Pavlovian to instrumental transfer test or to a devaluation test, she found evidence that the animals were not able to recall the unique stimulus outcome memories. The cues would come on, but they were ineffective in biasing their choice behavior during the pit test, and the animal would continue to go to the food port despite, in some cases, the cues predicting a reward that was no longer valuable. And so this suggested to us that whereas projections from the lateral OFC to the BLA might be important for encoding information about rewards, projections from the BLA back to the lateral orbital frontal cortex might be important for retrieving these memories, at least when they're stimulated by a stimuli in the environment. We also found that these lateral OFC to BLA projections are not necessary for retrieval in that experiment. The BLA also projects back into the medial orbital frontal cortex. There's some nice tracing work from Nina to convince you of that. And these projections, things start to get a little bit interesting. We inactivate these projections, and we find actually no deficits in Pavlovian to instrumental transfer. When Nina found this, she brought it into my office, and she said, it's the most beautiful pit I've ever seen, but it's not the effect that we were hoping for. We were kind of thinking we'd turn off these projections, and they wouldn't be able to retrieve the stimulus outcome memories. And that turns out not to be the case. So this tells us that despite the fact that we've inactivated BLA projections to the medial OFC, they know the stimulus-reward relationships, they know the action-reward relationships, they can even put that information together. But then when she went on, just like a week later, to do the devaluation test, here she saw a really robust deficit. Although we have evidence that they know they're able to retrieve the unique stimulus-outcome relationships, they aren't able to update their conditional responding, they're going to the food port, based on the value of the outcome that's predicted, which is suggesting to us that whereas projections from the BLA to the lateral OFC may be important for encoding or retrieving stimulus reward memories themselves, the value component of that memory, the stimulus predicts a specific reward, and that reward has a value right now, that value component may be disseminated from the BLA back into the medial orbital frontal cortex. So let me sort of put it all together for you, and it's a conclusion, but really it's a hypothesis we're working on moving forward. Our data suggests to us that the basolateral amygdala is important for encoding information about rewarding events, their value, their relationship with stimuli in the environment, and that the lateral OFC is supporting the BLA in this function. It's sending some signal into the BLA that is crucial for its ability to encode information about a reward's value as well as its relationship to stimuli in the environment. The medial orbital frontal cortex, we think, may be sending some information into the BLA about the current state, that state being defined by external stimuli as well as internal stimuli, to retrieve the right reward memory given the circumstances. And those memories we think may in part be stored in the BLA and disseminated back to a variety of regions, including the orbital frontal cortices themselves, where the BLA may be helping the lateral, ventrolateral, orbital frontal cortex to know what rewards can be expected in the current state, given your current set of circumstances, and that the value component of that memory might be submitted back to the medial orbital frontal cortex, perhaps for use in comparison with other potential rewards that might be available, and that these regions are then feeding into a broader decision-making network. So this sort of reward memories in the BLA going back to the orbital frontal cortex for use in making decisions. Said in sort of a different way, the orbital frontal cortex and the basolateral amygdala, these circuits are enabling the encoding and retrieval of reward memories to promote adaptive reward pursuit and decision making. But I think our data have revealed that there's a lot of dissociability in the system. I didn't spend too much time talking about it, but I did show you some evidence that we can either prevent or cause reward value encoding without having any effect on the best we can get the emotional response of the food reward, its palatability, indicating that there's some dissociability between how palatable the animal found this food reward, how pleasing he found the reward to be, and how valuable he then thought it was and then used that information in the future. These two things can be dissociable. We've also found some dissociability in the encoding, the circuits that are important for encoding information about rewards, versus those that are important for retrieving those memories under the right circumstances. And if this dissociability exists in the system, it creates a vulnerability in the brain, whereby things could become dysfunctional, and this could create bad decision making. And to this end, I'll just show you one kind of cluttered slide, but it makes one point, which is that we've been investigating this dysfunctional reward learning, and in particular, we've been investigating it in withdrawal following chronic opioid exposure. Essentially, what we found is that if you give animals a whole bunch of morphine and then you let them go into spontaneous withdrawal, the way in which they assign value to rewarding events, in this case I'm showing you for food rewards, we've also done it for water rewards, other groups have done it for opioid rewards themselves, becomes dysfunctional. They assign an inflated value to the reward such that they will pursue it with greater vigor than controls. They'll do so despite the fact that it's maybe not adaptive, given the fact that maybe they're sated or something like this. They really shouldn't be working for sugar. And this will disrupt the normal correlation that exists between how palatable the animals find the reward and how vigorously they pursue it. And so we've been suggesting that perhaps dysfunction in OFC BLA circuits, top down, bottom up, may be a potential pathway to addiction and maybe some other mental illnesses. And essentially this is something that our group is working on moving forward. So with that, I will remind you of who did this work. One is Melissa Malvaya. She's a project scientist in my lab who's on the job market this year, and she is absolutely phenomenal. Nina Lichtenberg also headed up a good bit of this work, Ash Morris, and we had a slew of really talented undergrads and technicians in the lab who helped us a lot with this. I'll thank our collaborators and our funding sources and also you for your attention.